The purpose of this video is to explain how we extract energy from food molecules like glucose into ATP, whether that's through aerobic respiration, fermentation, or other metabolic pathways. Let's start with the basics. Energy is stored in glucose, right? In the chemical bonds, the carbon-carbon bonds, carbon-hydrogen bonds. We're going to extract that energy and store it in new bonds of ATP. Remember, ATP is made from adenosine diphosphate. We add to that another phosphate, and it becomes adenosine triphosphate with all three of those phosphates. That's where the energy is stored. We're doing that partly because there's too much energy in glucose, 686 calories, and we want to get it down to something a little more manageable, somewhere around 9 calories, something that the cell can handle to do little tasks. In the case of aerobic cellular respiration, this happens in 3 stages: glycolysis, Krebs cycle, and electron transport system. Remember, these are stages, they're sets of chemical reactions. The first one, glycolysis, happens in the cytoplasm. The next 2 happen in the mitochondria. Keep it in mind that the main goal is to produce fully charged ATP molecules, and as many as we can. There are going to be a lot of byproducts and intermediates along the way. So let's track those. Let's put all the inputs on the left side and the outputs on the right side. Let's start with glucose and trace that all the way through. So glucose is a 6-carbon molecule shown as these 6 black dots. That goes into glycolysis, and what comes out are 2 pyruvate molecules. And those are 3-carbon molecules. So basically, we've broken glucose in half. That pyruvate then gets fed into the Krebs cycle. It's converted to acetyl-CoA, goes through another variety of steps, and is given off as carbon dioxide. All right, so now our glucose is gone, and that's it. Let's go back step by step and look at the energy-containing molecules that we got out of it. Let's start with ATP, since that's mainly what we're after. Glycolysis is a little bit strange in that initially we put in 2 ATP. I know we're trying to get ATP out of it, but that's what we put in, and what we get out is actually 2 ADP, adenosine diphosphate, and their phosphates. So that's actually been broken down. This needs to be recharged. But we also then put in 4 ADPs, the uncharged versions, and we get out of it 4 ATPs. So a lot of times we say net, we get 2 of these guys, right? Just 2. We spent 2, we got 4, so we ended up with 2. Anything else in here? Yes, one more thing. We put in 2 NAD+. This is a type of nucleic acid. It is an electron carrier. Those go into glycolysis. They help extract some of that energy, and they come out as NADH. Again, 2 of those. For the Krebs cycle, I'm going to assume that we're doing this for one entire glucose, so we put in 2 pyruvates. What we also have to put into that is 8 NAD+ molecules, 2 FADs, and 2 ADPs with their phosphates. All right, so those all go in to the Krebs cycle, and as those reactions occur, extracting energy from glucose, what we get out is 8 NADHs, 2 FADH2s, and 2 ATP. So 2 more of these guys. Not really a whole lot on the way to our total, but the big payoff we know comes down here where we get 32 ATPs. Finally, the electron transport system. This works a little differently than the other ones. Obviously, our glucose and all that is used up. So what powers this is our electron carriers, all that NADH as well as the FADH2 CO2 that we've been producing. So we've been producing those have been outputs, but in this case, they are going to be inputs. So that's where our electrons come from. They travel through this electron transport system. That produces a hydrogen ion gradient. That gradient is used to drive ATP synthase, which makes lots of ATP. Now we do have matter in, matter out, so we're also putting NAD plus molecules out here, as well as some FAD. That's essentially the uncharged version of those guys. The other thing that needs to go somewhere is our electron, and so that's where all those oxygen molecules come in. Oxygen is going to go through this system, accept those electrons, along with some hydrogen ions in that system, and is going to come out as good old H2O. So there's the rest of the matter that goes in and out of this thing. Before we leave aerobic cellular respiration, let's put this into context. First, all that ATP, we just made it all. Where does it go? Well, it drives muscle contraction, protein synthesis, DNA synthesis, sodium-potassium pump, calcium pump. It's what does the powering of the work inside of your cell. What about the rest of it? Glucose. Well, this process is happening inside the cells of your body, but glucose is coming in your body by eating it. Oxygen is another necessary input that comes in your body by breathing in, and it gets into your cells by what? Simple diffusion. Water probably just stays in the cell. If there's too much, it would leave the cell by osmosis. Carbon dioxide. Carbon dioxide is exiting the cell by simple diffusion, and then out of the bloodstream into the lungs by simple diffusion as well. Most of the rest of the materials, all these NADHs, FADs, ATPs, and ADPs, the whole alphabet soup, mostly just keeps getting recycled within the cell, kind of rides this merry-go-round again and again and again until those molecules are finally broken down, and then they get recycled in a different way. Moving on to fermentation. We'll just treat the case for humans where we do lactic acid fermentation. Some bacteria do these as well. I just have kept some of the key details here. Remember that glucose goes in, gets broken down into two pyruvates. We also need NAD plus as part of that, which produces NADH. Now, when do we use fermentation? We use it when we're out of oxygen. Where does that come in? Down here. We need oxygen to keep this running. Well, if we don't have oxygen, then this can't run, which means, one, we can't get all that ATP, but also we have no way of getting rid of this NADH. Why is that a problem? Because normally this is where it goes to produce the NAD plus that we need to run glycolysis. I know, it seems convoluted, doesn't it? But it's a real monkey wrench in the works of this machinery. Since all this is happening in the mitochondria, also means the Krebs cycle can't happen because it also produces lots of NADH and has nowhere to recycle it. Needs to have NAD plus. So this doesn't happen immediately when oxygen levels run low, but if they're low for long enough, then that's what happens. The solution, fermentation. What is that? Well, remember these are electron carriers. I'm going to put those two little dots there to represent the electrons being carried by this molecule. I'm going to give those electrons actually to pyruvate, and when I do, then it's going to actually form lactic acid. It's really not much of a big change chemically. We're going to add some electrons, which are going to change some of the bonds. It's going to add another hydrogen on there, take a slightly different form. It's still going to be a 3-carbon molecule, which the liver will eventually recycle for us. And what that allows is that NADH, now that it's given up its electrons, can be reformed into NAD+. That means glycolysis is happy, it's complete, it can keep going and give us even just those 2 measly ATP out of the process. But it's better than nothing. All right, what about those other metabolic pathways? Well, really, all of this is a metabolic pathway, right? We have just been using the metabolic pathway for glucose to be broken down through this path, often a very convoluted path, to produce ATP. So that's its path. And this is true for anything that's related to glucose, like starch or glycogen. Those get broken down into glucose, right? Because they're just made of glucose monomers. And once they do that, they can start in glycolysis and back up from there. Okay, what if you don't have glucose, or if you don't have enough, or if you want to use something else? We can do that. We know we can use proteins and lipids for our calories as well to produce ATP. How would we do that? If we don't have glucose, then we'll probably next turn to lipids. Those lipids, if you remember, are really long hydrocarbon chains. So at least the fatty acid part is usually somewhere on the order of 14 to 20 carbons long. So I'm just drawing as these little dots, kind of like the glucose was drawn. So they're really long. Obviously, that can't be fed into glycolysis. It's the wrong molecule, wrong fit. The enzymes won't recognize it. It can't do much with it. So what I do is a process known as beta oxidation. Sorry, that's oxidation, which takes and cuts up those fatty acids into little 2-carbon pieces. All right, so I have a whole bunch of 2-carbon molecules. Well, remember what I got out of glycolysis was 2-pyruvate, and which is a 3-carbon molecule. I converted that into acetyl-CoA and then went through the Krebs cycle. So that means lipids can be fed into the Krebs cycle. But if you remember from the bit about fermentation, the Krebs cycle is not going to work for me since it's in the mitochondria along with that whole electron transport system unless I have oxygen that's allowing this to work. So we can use lipids, but we can't put them into glycolysis. We can feed them into the Krebs cycle, which only works if we have oxygen. If I'm not using glucose, and if my body doesn't want to use lipids or doesn't have enough, then it's going to turn to amino acids. Now, amino acids have a particular structure to them. So here's one example of amino acid. Everything we've dealt with before has carbon, hydrogen, and oxygen. That's the case for glucose and lipids, and that's really all these processes are going to accept is things like that. So one of the first things we do, and this happens in the liver before it even gets to your cells, is to take off this amino group, which forms ammonia. The body actually takes that, combines it with carbon dioxide, and forms urea, which we get rid of in our urinary system. So now we've dealt with that part of it, and now we have a compound left that looks somewhat familiar. Here we can take this 3-carbon molecule. And remember, each amino acid is a little bit different. Some of them have 3 carbons, some have like 7 carbons, others just have 2. But if they have more than 3, then they're going to be chopped up into probably 2-carbon pieces, and most of them are going to be fed into the Krebs cycle again. Same thing applies as before. We can't use glycolysis. It doesn't really apply. None of those enzymes are going to recognize most of these molecules anyway. And the Krebs cycle is only going to run if it has somewhere to give up its NADH molecules to, which means we need oxygen present. So if we have oxygen, then we can use amino acids as an energy source to make ATP down here. Obviously, we're not going to make this ATP because that came out of glycolysis, which we're not using.